Welcome to the Deep Dive, where we take complex source material and really pull out the essential breakthroughs you need to know about right now. Today, we're moving past all the general hype around AI. We're focusing entirely on the concrete, groundbreaking tools that are being built in labs as we speak and how they're just fundamentally changing the pace of scientific discovery.
And it's so necessary. I mean, the core challenge in biomedical discovery today is just it's information overload. We're drowning in data. Drug development is incredibly resource intensive. It's expensive. And part of that is because, we've already picked all the low-hanging fruit.
The easy targets are gone.
They're gone. So to tackle the new, really complex targets in things like cancer or fibrosis or rare diseases, we need AI systems that do more than just process data. They have to manage it, interpret it, model it, and most critically, reason across these huge, disparate data sets.
And that, for you, is our mission today. We've synthesized findings from some really cutting-edge papers on everything from colossal knowledge graphs to specialized molecular folding, even chemical reasoning benchmarks. We're going to unpack 4 distinct but interconnected pillars of AI innovation. We've got knowledge graphs, autoencoders, conditional diffusion models, and sophisticated LLM reasoning that together are creating a critical shortcut to discovery.
These are the technologies that are turning scientific uncertainty into actual, actionable insights. And we should probably start where all scientific progress begins, which is organization.
Okay, let's unpack this. The first, foundational step in accelerating discovery is just organizing the world's fragmented, enormous body of scientific knowledge. This is where the iKraph project comes in. So when we talk about AI as the ultimate librarian, pulling insights from the entire scientific corpus, what does that scale actually look like?
The scale is, well, it's simply vast. The iKraph Knowledge Graph, or KG, processed over 34 million PubMed abstracts.
34 million.
34 million.
Yeah.
Representing decades of scientific publishing right up to May 2023. And after processing all that language, it pulled out over 10 million unique entities. So things like genes, drugs, diseases, and then more than 30 million unique relationships between them.
10 million entities and 30 million connections. I mean, that's already enormous. But what makes this specific KG more powerful than just, a really, really big database?
It's the integration. That's what sets it apart. It isn't just a library of abstracts. It also pulls in curated data from 40 other public biomedical databases. So it's like a unified, massive map where every concept is connected to every other known concept from both published text and structured biological records.
And these AI tools are only as good as the data they eat, right?
Right.
So quality assurance here has to be essential. How did the researchers even confirm that the AI was extracting these millions of connections accurately?
Right, and that's where the rigor comes in. The pipeline was validated by competing against, human annotation standards in these formal challenges. It won first place in the NIH Litcoin NLP challenge. With a really respectable score of 0.7186, and it also won another big one, the BioCreative Challenge.
All right.
But critically, manual verification later confirmed that the AI's accuracy was basically at the level of a human annotator. So it's not just fast, it's trustworthy.
Trustworthy and huge. Okay, so once you've built this massive verified library, how does the AI actually use it to connect things that scientists maybe didn't realize were connected? It doesn't make discoveries on its own.
That's the key innovation. It's called probabilistic semantic reasoning, or PSR. This is the core mechanism that lets it do causal inference. So instead of just pulling a direct A to B relationship that was already written in a paper, PSR lets the system infer indirect relations. A is related to B and B is related to C, so maybe A is causally related to C, even if no single researcher ever published that specific link.
So it sounds like it's using logic, maybe not biological logic, but logical connections to bridge these huge distances in the data.
Precisely. And the huge benefit of PSR is that it's highly interpretable. You can trace the reasoning back through the chain of direct relations that the AI use. It doesn't just give you a black box answer. It actually shows you its work.
Let's talk about the real-world impact of that, specifically on drug repurposing. The motivation there is crystal clear. New drug development is crazy expensive and slow. Finding a new use for an old safe drug is, well, it's the holy grail.
Absolutely. And they showed this power retrospectively with COVID-19. They basically turned the system loose in early 2020. Between March and June of that year, the method identified a whole bunch of viable candidates for repurposing.
Okay.
And then they tracked those predictions. And they found that 1/3 of the drugs that identified in those first 2 months were later validated by actual PubMed literature or clinical trials. That shows real-time pandemic-level responsiveness.
1/3 validation in real time. That is a massive hit rate. But the timeline reduction for the chronic, slower-moving diseases is even more stunning to me. It just highlights how inefficient human-only research can be.
Well, look at cystic fibrosis. Historically, when a drug was proposed for CF, it would typically take somewhere between 2 and 33 years to get validated.
33 years.
A median time of 9.4 years. The iKraph study showed that if its predictions were acted on immediately, it could hypothetically slash that median validation time down to just 2 years.
Hold on, so you're telling me this technology could potentially take a research path that lasts a decade and just collapse it into 2 years with a high degree of confidence?
That is exactly the implication. I mean, it changes the fundamental economics and ethics of drug development.
Yep.
We are talking about saving nearly a decade in that most critical early discovery phase. It lets researchers move straight to the expensive, targeted clinical testing with high probability candidates. This is how you fundamentally accelerate medicine.
That sets the stage perfectly. So we've organized the knowledge, but once we have it, we still have to handle the complex, layered reality of biological data. The multi-omics challenge. We're talking genomics, transcriptomics, proteomics, all generating these vast, complex, multi-dimensional data sets. This is where it gets really interesting for me, the AUTOENCODIX framework, which is designed specifically for this.
Yes. AUTOENCODIX is this unified open source framework built on PyTorch, and it standardizes training autoencoders on large-scale biomolecular data. You can think of it as a generalized platform for what we call representation learning. It supports all these different AE architectures like VanillaX, VerX, StackX, so researchers can pick the best tool for their specific kind of omics data.
The basic idea of an autoencoder is you're teaching a machine to compress high-dimensional data into a smaller, meaningful, latent space and then decompress it backward.
That's it.
But what I find fascinating here is the focus on interpretability. So often, that latent space is a complete black box. But one of their architectures, ONTIX, is engineered for transparency right from the start.
That is the breakthrough with ONTIX, the ontology-based VAE. It achieves what they call explainability by design. Most AI models, they require this tedious post hoc analysis to figure out what biological features they even focused on. ONTIX avoids that by giving the AI a blueprint before it starts.
How did that work in practice?
They restrict the model structure. Specifically, the decoder's feature connectivity has to strictly conform to a known biological ontology, like the reactome pathways.
Ah, okay. So it's like giving the AI a very detailed pre-labeled map before it starts drawing. It's not allowed to draw anything outside the lines of known human biology.
That's a perfect analogy. And the trade-off is knowledge over raw, unrestricted precision. In one case, they set the latent space dimension to 29, which corresponds exactly to the number of top-level reactome pathways. The model knows that if it compresses the data, every single compressed dimension has to correspond to one of those 29 high-level biological functions.
So they're deliberately diminishing the model's raw reconstruction ability compared to an unrestricted one. But that drawback is totally justified because every single piece of information it outputs is guaranteed to reflect a known biological pathway. It's counterintuitive, but brilliant.
It ensures the model's compressed representation is inherently biologically meaningful. It makes the results directly applicable for generating new hypotheses.
And speaking of application, the ability to translate data from one modality to another, that sounds incredibly useful for labs that might have, say, proteomics data, but no genomics data for the same sample.
And that's managed by XModalix, the cross-modal VAE. This architecture, it learns separated latent spaces for different data types, but these spaces are mathematically aligned. This allows for really powerful data fusion. The source material cites a key application, omics to omics translation, specifically predicting gene expression, so RNA-seq, based on D-Day methylation data from cancer samples.
So it can essentially fill in the missing data points.
Exactly. And importantly, it can even handle unpaired multi-omics data. So if you have dataset A from 100 patients and dataset B from a different, maybe partially overlapping set of 100 patients, XModalix can still build the translator. And that dramatically increases the utility of messy, real-world data.
We've gone from structuring knowledge to interpreting complex data layers. Now we move to concrete physical structure. We all know AlphaFold 3 revolutionized protein prediction. It made headlines for modeling complexes between proteins, nucleic acids, small molecules. But SiteAF3 shows how focusing on site-specific folding can give you results that are even better, especially for drug designers.
It's a classic scientific principle, right? Generality is great, but specificity wins in applied fields. CiteAF3 is a novel conditional diffusion method, and its core innovation is refinement. So instead of trying to model the entire complex from scratch, which is computationally expensive, CiteAF3 fixes the receptor structure.
And why is fixing the receptor structure so critical?
Because in drug discovery, you already know your target protein's structure. You're only worried about the small molecule, the ligand, and how it binds into a specific pocket. By fixing that receptor, CiteAF3 just massively constrains the problem's space.
Right.
It lets the model dedicate all of its computational energy to a much more targeted, focused exploration of how that ligand fits inside that one specific binding pocket.
So it's like saying, we know the landscape and the destination, now just find the absolute best parking spot for this one little car.
Yeah.
What kind of quantitative edge did that give them? Especially for small molecules, they're notoriously tricky.
The performance gain on the FoldBench dataset was substantial. CiteAF3 got an accuracy of 69.7%, which was a notable jump from AlphaFold3's 62%. But the most impressive metric is about precision. The best CiteAF3 model reduced the median ligand root mean square deviation, or LRMSD, by 30.9% compared to AF3.
Okay, for those of us not building molecular models every day, what does reducing the LRMSD by 30% actually signify?
Right. LRMSD is just the measure of how accurately the predicted placement of the small molecule sits compared to the actual experimental placement. Reducing it by nearly a third means we're getting a level of structural certainty that was, well, it was unheard of for these tricky, flexible binding pockets. In drug discovery, this is the difference between a viable candidate that actually fits the lock and a false positive.
That improvement is massive. And it sounds like the focus was really on improving the most challenging cases.
It was. CiteAF3 really fulfilled its objective by significantly boosting accuracy for the bottom 20% of structures. The ones where the original AlphaFold3 prediction had the lowest confidence scores and were, the least useful to researchers. It turns unusable predictions into potentially viable drug candidates.
And we shouldn't overlook its versatility beyond small molecules. It seems to excel wherever flexibility is a big issue.
That's right. For peptides, which are just notoriously flexible, CiteAF3 showed a mean DocQ score that was 11.2% higher than AF3. And for maybe the most difficult of all, protein-RNA complexes, the median DocQ score saw a huge rise of 44%. It's succeeding exactly where generalized models tend to struggle.
Plus, focusing on just the site offers computational benefits, I have to imagine.
Definitely. The conditional approach means a reduced GPU memory footprint and a significantly faster runtime, especially when researchers can input known pocket and hotspot information. You get better results, faster and cheaper. It's the ideal combination.
Okay, so we've structured the knowledge, translated the data, and modeled the molecules with precision. The final piece of this puzzle is chemical reasoning. Can AI actually think like a human chemist? Can it solve problems that require inference and analysis, not just data retrieval? The ChemIQ benchmark gives us an answer.
And this benchmark was so crucial because it established a really clear reasoning gap. Earlier, non-reasoning large language models, they struggled. They achieved only 3 to 7% accuracy on core chemistry tasks.
3 to 7%, that's basically random guessing.
Pretty much. And it confirmed the prevailing view that LLMs were just fundamentally incapable of true molecular comprehension.
But then the introduction of explicit reasoning models like O3 Mini and the really sophisticated Gemini Pro 2.5 produced this just stunning, dramatic leap forward.
Stunning is the right word. These advanced models achieved a 50 to 57% overall accuracy on the 816 questions. And the critical finding wasn't just the score, but how they did it. The increased reasoning levels significantly boosted performance, and their chain of thought, their step-by-step process, it often mirrored the structured reasoning of a trained human chemist.
Let's break down some of these tasks. Molecular comprehension, for instance, understanding the internal structure.
Okay, so take the task of determining the shortest path between two atoms in a canonical smile string. For the non-chemist, a smile string is just a one-dimensional text representation of a molecule. Solving this requires the AI to, computationally at least, convert that string into a 3D graph and then run a search on that graph.
That's a huge leap from simple text processing.
It is. The top reasoning model, O3 Mini High, achieved 89% accuracy on this, and the analysis of its reasoning strongly suggested it was actually constructing an internal graph representation and performing that search. It's a level of structural understanding we just thought was impossible for this class of AI.
What about translating chemical notation? That used to be a total failure point. Converting a smile string to the formal IUPAC name is like translating very specific code into descriptive text.
Historically, smiles to IUPAC naming gave you near zero results for LLMs. It just couldn't do it. Now, these reasoning models showed meaningful success. Gemini Pro 2.5 scored up to 44% accuracy. And while 44% isn't perfect, going from near zero to nearly half correct on a task that needs strict adherence to rules, it's a monumental shift.
Okay, finally, we get to the pinnacle of chemical reasoning. Structure elucidation from spectroscopic data. This is what you do in organic chemistry when you're handed raw data and told, figure out the unknown molecule.
The NMR elucidation task is the true test. Nuclear magnetic resonance data, especially 2D NMR, gives you these complex constraints on how atoms are arranged in space. Gemini Pro 2.5 correctly generated smile strings for small structures up to 10 heavy atoms with accuracy between 73 and 94%.
But the ultimate test cited in the source is the complex structure. That's the kicker.
It is. The most critical finding was that Gemini Pro 2.5 successfully solved the complete structure of one complex molecule with 25 heavy atoms using multidimensional 2D NMR data.
Wow.
Solving a structure that complex requires synthesizing numerous, often contradictory data points into one single coherent molecular structure. This just definitively shows advanced high-level chemical reasoning in these models. The AI can now interpret spectroscopy.
This has been a fascinating deep dive. I think it really demonstrates that the AI revolution in biomedicine is no longer speculative. The common thread here is that AI is moving from being a passive data filter to an active scientific co-pilot. We've covered how it's structuring knowledge with iKraph, translating complex multi-omics data with autoencodix, modeling molecules with site-specific precision using SiteAF3, and now demonstrating genuine chemical reasoning with ChemicQ. This convergence gives researchers tools that are faster, smarter, and just fundamentally restructure how we tackle the hardest biological and chemical problems.
And this convergence raises a really provocative thought for you to consider. The source material for SiteAF3 mentioned that one of the key challenges for models like AlphaFold isn't necessarily better folding algorithms, but developing more effective scoring or ranking algorithms to pick the best structure out of millions of possibilities. So, given that AI can now interpret and reason through complex spectroscopic data like 2D NMR, could the next major breakthrough in structure prediction come not from improving the folding models, but from training LLMs to become the ultimate chemically informed ranking experts? Could the AI that tells us if a structure is even chemically possible be more valuable than the one that generated it in the first place? That is the fascinating direction this whole field is heading.